I work for Affinity Biomarkers. Affinity formed out of the London Hospital University system, and we're a translational medicine company. Our aim is to actually take ideas from academia and SMEs and help them translate through those development pipelines that have just been highlighted. So we deal from D4 through to T4, that type of area. We were initially based in Imperial College in London, and we're planning on opening up a lab which should have been up and running by now. But of course, we ran into the problem that was COVID, and that put us huge delays on our expansion plans and moving in labs just for logistical purposes. We're involved throughout the UK, currently with multi-centered COVID trials, including biobanking, proteomics, and metabolomics of COVID and long COVID. So Affinity's aims are to assist, as I say, academics and SMEs and companies to validate their biomarkers and make sure that they can get them into the right place at the right cost. We support academia, biopharma, and with clinical trials, and our expertise is both in the analysis, but also in facilitating, pulling together the larger clinical trials that people need. Our mission, of course, is to make sure that you guys can get what you need when you need it, and at a price that is favorable, of course. So, of course, around that we have to consider quality. We comply to the Human Tissue Act. We're in the process of 15189 ISO accreditation. We've got a quality management system. We track all the samples that we handle, and we have an alliance with Biocrates for doing targeted metabolomics on large scale. So the structure of Affinity is the clinical chemistry and immunoassays are based down at our new lab in London, and the mass spectrometry lab will be based up in the Manchester facility, and hopefully will be moving forward at a speed once the opening up from COVID occurs. So the Manchester mass spec lab will have standard assays for metabolites, for drugs, and we'll also be setting up a targeted and possibly a non-targeted metabolomics facility there to meet all the needs, all your needs from discovery through to validation. So that basically is the sales pitch over, and I decided, as I say, to pitch at quite a low level. And quite often when I meet with clients, I have pretty much the first meeting is the same conversation over and over again. And that's because basically we're divided by a common language. The meanings of certain words and certain implied understanding between biologists, clinicians, and analytical chemists, which I am, mean that we can, you can ask what you think is a relatively simple question, and I go away and give you a complicated answer, and I tell you that your analysis is going to cost a lot more than you think it should do. And the usual questions that I'm asked is how much of my compound is in this sample, or what is in this sample, and then the obvious question of how much will it cost to tell you that. The idea, if we look at the first question of how much compound is in the sample, we've got to pull out what we mean by how much. So to an analytical chemist, how much is a a question which can be answered in multiple ways, from is there any of the compound in the sample at all, through to is there more of that compound in this sample versus a standard or a different sample. Do you want to know exactly? Do you run it with a calibration curve? Do you want more precision? Because of course, all the measurements we make have errors built in. And I'll talk more about errors a little bit later, but to mitigate some of those errors, we can either use what are known as surrogates, which are compounds that are similar to the analytes, but structurally a little bit different, or we can use isotopically labeled standards. Of course, as we go down this list, the cost increases, the accuracy which increases, usually the time to develop the methods increase, and of course the cost increases, which is something that's important for you to remember, especially if you have thousands of samples you want to analyze. So, some common sources of errors: the biology itself in living systems, variability, especially in compounds that are non-homeostatically controlled, can vary up to 30%. There's collection and storage, both absorption, losses in sample storage, be it in a fridge or a freezer. One example I have of losses due to storage is I was running an assay that relied on measuring of acetate, and we, as was common practice, shoved the samples in the freezer over the weekend and reanalyzed some just to ensure that the levels were coming out the same, and lo and behold, they weren't. It turned out that freezing the samples pushed the acetate out of the solution, and of course, it then being volatile, it evaporated from the sample and was giving false readings. And it turned out that for storage of these samples with acetate, it was important to actually refrigerate the samples, and subsequent testing showed that these could last for over a week in the refrigerator, whereas a simple freeze thaw would destroy the accuracy that we achieved for the samples from the freezer. There are systematic errors, miscalibration of pipettes, errors in the hardware that are repeatable, and then there are random errors, which are the user errors, the missing of an air bubble in a pipette, and so on and so forth. So we also would like to know what you want to know about your analyte. And chirality in biological systems can be very important. Molecules that have a reflection or a rotational symmetry can behave very differently in biological systems. So it might be important to you to know which enantiomer or stereoisomer you have in your system. If you want to know this, of course, it complicates the system. And the last thing your analytical chemist would want is to report back to you a bulk analytical result and it not be the result that you want. We also want to know about the physical properties in the case of acetate, the volatility, if the samples there that are stable, or the analytes are stable in the samples. If you want to analyze ATP, you've got to take special precautions and analyze them quite quickly. Will the sample ionize in the technique that, or it may dictate the technique that we have to use if the sample is not suitable. So for later I talk about HPLC or mass spectrometry. If it doesn't analyze, we might want to use HPLC. If it does ionize, we might want to use the mass spec. The other point, which is for the safety of the analyst, is the sample does it contain hazardous materials, which may be, of course, generally speaking, having worked in this field for too many years, we always treat everything as if it's hazardous, but it's always nice to know if there are any known hazards, including infectivity, such as from COVID or HIV or the like. So it's important that a assay should be fit for purpose. And the reason I put thin layer chromatography here is I had an assay where a biologist walked up to me and said, I want to know, is there any, how much of a compound is in my sample? I have 10,000 samples. And with a quick back of the envelope calculation, I worked out that it would cost him about 500,000 to have a quantitative assay developed for and run for those large number of samples. But what having talked to him, he actually didn't really want to know how much was in his sample. He wanted to know which samples had any of the compound in. And that's an easier question to answer. And what I recommended to him was that we bought some TLC plates. I taught him how to do TLC. And within a week, he managed to go through his sample set using a simple, cheap, rapid, efficient, and easily scaled assay, and all for the cost of about £50 for the cost of the box of TLC plates. Again, saving money, we can analyze things by HPLC, usually for semi or non-polar analytes. It can be coupled to mass spectrometry with care. Normal detection systems are UV, although you can have evaporative light scattering detection or refractive index if you want. If you
Just
want
a minute to go, Scott. Sorry to interrupt.
A minute?
You. Yeah.
I better hurry up.
Thank you.
Yeah. So mass spectrometry, the complexity depends on the accuracy that you want to use, and usually most people go for triple quadrupoles, which is the pharma standard for quantitative, and it also generally gives you the best linearity, so the widest dynamic range of measurements, because both the bottom concentration and the top concentration may be important to you. So understanding that what the range that you want to measure is also important, because there may be other methods that we can use to concentrate your analyte so we can get you the results that you want. So know as much as possible about your analyte, know what you need to know and what you want from your analytical purpose. Choose a fit-for-purpose method. It'll save you time and money. Ask advice from your analytical chemist. And as a general rule, try to keep as many things as possible constant, including the tubes that you collect blood in, the tubes that you store your samples in, and the number of free store cycles that your samples go through. Because all of this can affect your analytical results. And at that point, seeing as my clock now says 25 past, I'll stop sharing and I will take any questions if there are any.